This is the IGCSE Human Biology June 2019 Sample Assessment Paper 1, question number 6. So let's begin. Here, part A says that the diagram shows how a competitive inhibitor reduces enzyme activity. Here they have provided us a structure of an enzyme and a substance, which is basically the inhibitor, which is the competitive inhibitor that has fit into the active site right over here. All right. So, since the concept of enzyme inhibitor and enzyme activity has already been discussed, if you have seen question number 2, I have already discussed how these work. But since this competitive inhibitor has come again in this question, I will discuss this part in brief. So, basically, focusing on how the competitive inhibitor works. So let's have a short review on how the competitive inhibitor works. So inhibitors are basically substances that reduce the rate of reaction, enzyme catalyzed reaction. How they do this is basically they interfere with the work of the enzyme during the reaction. As you can see, this is the competitive inhibitor and this is the non-competitive inhibitor. There are two types. So let's focus on the competitive. What the competitive inhibitor do is that it has some sort of similar shape to the substrate, and what it does it is that it is going to settle down somewhere on the active side of the enzyme. So since it is blocking the pathway of the substrate to the enzyme, this means that the substrate is going to drift away because it is unable to fit to the active side of the enzyme, and hence as a result the reaction will not take place until this inhibitor moves from its place and allows the substrate to bind again. All right. So since we have discussed about it, let's move back to our diagram. So here is the diagram. As you can see, the same has happened over here, that same shaped, similar shaped inhibitor, like this active side of the substrate, not completely filling, but it has settled over here, filling the active side almost, so that the substrate now cannot enter and let the reaction happen. So basically, the effect of a competitive inhibitor makes reduce the enzyme catalytic reaction rate. However, there is one more, which is a non-competitive inhibitor that has a much stronger effect because that stops or holds the reaction at once. But let's focus on this first. All right. So part 1 says that explain why the competitive inhibitor is able to bind to the enzyme. Use the diagram to help with your answer. Okay. So as you can see over here, the enzyme, it has this active site, and the competitive inhibitor is able to bind to this place because it has a similar active site and a similar shape to the substrate, which means that it is going to be complementary to the enzyme also, and hence it can bind right over here to the active site almost. So since it is sitting over here, the substrate can no longer enter unless this has moved. All right. So let's answer this question. It is a 3-marker question, means we have to be careful that we are mentioning at least more than 3 points. So 3 points are over here. So let's answer. As you can see here, I have mentioned that the inhibitor is able to bind since it has a similar shape to the substrate. Therefore, it means that it is complementary to the shape of the enzyme and hence binds to the active site. All right. So since we are done with this question, let's move on to the next part. Part 2 here, it states that the graph shows how a competitive inhibitor affects enzyme activity. So as you can see over here, it shows a graph. The graph has on its x-axis substrate concentration and y-axis the enzyme activity in arbitrary units, which is the dependent variable. This is the independent variable. And here they have shown the effect of inhibitors on the enzyme activity. This slope is for the effect of enzyme activity with the competitive inhibitor, and this one is without the inhibitor. This is the maximum rate of reaction, the level. All right. So as you can see here that the graph with the inhibitor, this part, as you can see, is greater than that with the competitive inhibitor. Why? This raises a point that the competitive inhibitor is slowing down the rate of reaction. Hence, it is not reaching its highest point. And also you can see right over here, the graph levels off at the maximum rate of reaction earlier for without inhibitor, whereas with competitive inhibitor, it takes a very long time for it to reach the maximum rate of reaction. Because since the competitive inhibitor is creating a competition on the enzyme for the active side with the substrate, so the inhibitor, as you can see earlier in the diagram, as I had said, that this inhibitor settles down here for a while, halting the reaction for a while, like delaying it. So unless and until it has moved away, the substrate cannot fit in, which means the reaction will take place, but it will delay because of the inhibitor. As you can see its effect right over here. So here the question says describe how enzyme activity is affected by the presence of a competitive inhibitor. It is a two-marker question, so let's answer this. As you can see, I have answered over here. Since it is a two-marker question, I have listed in points that number one, it lowers the rate of enzyme activity. Number two, it takes longer time to reach max rate of reaction for the competitive inhibitor. As you can see right over here, as I have explained. So since we have completed this question, let's move on to the next part. Here is part B. It says that the diagram shows another type of inhibitor to an enzyme. It is, okay, so another inhibitor that is settling on the shape of the enzyme, which is the body of the enzyme instead of the substrate. So the inhibitor affects the enzyme in different ways to the competitive inhibitor. Describe the differences. So if you have remembered, I said that there are two types of basic inhibitors. One is competitive and another one is non-competitive. So let us move back to the discussion phase. As over here, we have two types of inhibitor. One is a competitive and one is a non-competitive inhibitor. So as you can see over here, this one matches with the diagram in the question. This one was for the diagram, which is the first one, which is the competitive inhibitor. Here for the non-competitive inhibitor, as you can see. What this type of inhibitor does is that it is going to bind to somewhere on the body of the enzyme, and hence it is going to change the entire active site shape of the enzyme. So as we know that enzymes are specific and have its own specific substrate to fit into its active site. So if the active site is already destroyed, it means the substrate can no longer fit. It means the enzyme has gone useless and it cannot involve in any other reaction anymore. All right. So this is a very dangerous type of inhibitor for the enzymes. All right. So let's move on to the question. Back to the question. As you can see, it said that the inhibitor affects the enzyme in different ways to compare to the competitive inhibitor. Describe these differences. So as you can see that this inhibitor directly changes the substrate, I mean the active site, so that the substrate can no longer fit anymore. It cannot work anymore. All right, and for the competitive inhibitor, what it did is that the inhibitor was going to come and settle down somewhere on the active site because it has the same shape. Also, the inhibitor, which is a non-competitive inhibitor, does not have the same shape as the substrate as the competitive inhibitor had, so that it will not fit into any active site. It settles down in the body of the enzyme, changing the shape, which a competitive inhibitor does not do. And hence, it halts the reaction completely, whereas competitive inhibitor, it delays the reaction but does not halt the reaction completely. So since it is a three-marker question, let's answer this. So since I have discussed and also here I have listed down that these are differences, that this is a non-competitive inhibition and it binds at a place on the enzyme other than the active site and it changes the shape of the active site. Okay. All right. So I've answered this question. Let's move on to the next one. Moving on to part C. Here, part C says that a pie chart shows the worldwide industrial uses of immobilized enzyme. Here, they have provided a chart ratio of the proportions of distribution. They have biological research, diagnostics, and food and beverage, fabric and household, biofuel, and others. So they have all the proportions over here shown clearly by a chart. Part 1 says that the total estimated market value of industries that use immobilized enzyme is 3.9 billion. Okay, and calculate the estimated value of immobilized enzyme in the food and beverage industry. So basically, they're focusing on this part of the proportion. So since they have given us all of these values, so we can find out this part of this proportion by subtracting all these values from 100 at once. Okay, so let's solve this because we need to obtain this value to find the estimated value of the food and beverage. Hence, we're calculating this part. So let's do this. As you can see here, I have solved. I've calculated the percentage of food and beverage by subtracting all these values from 100 and it comes 21, which means that this proportion is 21%. So to calculate the estimated value of immobilized enzymes in the food and beverage industry, what I'm going to do is I'm going to take 3.9 billion over here, which is the total amount, and take 21% of it, which is multiplied by 21 divided by 100 due to the percentage sign, and it comes this value. All right. So here we have calculated the answer of the estimated value of immobilized enzyme in the food and beverage industry. It is a two-marker question, a very easy way to gain marks. And always remember that whenever in this type of graphs, whenever all of the proportion ratios are given, except for one, if they are, have asked you, if they have asked you to calculate the last proportion, you can always do this by subtracting all of these values from 100. Hence, you'll get this part. Okay, so let's move on to the next part. Part 2 here states that the statement about the use of immobilized enzymes in the food and beverage industry. Number one, amylase is used to convert lactose to galactose and glucose in the production of lactose-free milk. And glucose, number two, that glucose and fructose are produced from sucrose in the production of slimming foods. Okay, so which of these statements are correct? So they have given us option over here. So option A is 1 only, or option B is 2 only. Option C is both statements 1 and 2, and option D is neither of the statements. So as we know from the concept of immobilized enzyme, the answer is going to be B. Here I have marked it that the answer is going to be B. So it's a very easy one mark MCQ that glucose and fructose are produced from sucrose, which is true in the production of slimming foods. Okay, so moving on to part D. So this is the last part of the question. It says that describe how immobilized enzymes are used to detect glucose in urine. So this is a 4-marker question. It has asked us how immobilized enzymes are used in detection of glucose in urine. Okay, so before we start to solve this question, let's see one more piece of information. This is a part of your textbook, and information I have obtained from this textbook that for testing glucose in using the urine, what we can do, what they normally do is that they have biosensors which use test strips over here, as you can see. So these test strips are basically having immobilized enzyme bound to them right over this place. So whenever these strips are made into contact with the urine sample, or probably the urine sample is they're dipped either, or some way this urine sample is transferred to this stress strips, they change color. As when this happens, glucose in the urine is changed into gluconic acid and hydrogen peroxide right over here. As you can see this reaction. Okay, so due to this reaction, the color changes occur, and according to the color change, we can detect the level of glucose. So since they have asked us how we can detect the level of glucose, so let's answer that question. So as you can see, I have answered here. Since it is a 4-marker question, I have mentioned 4 points. Number 1, immobilized enzymes are bound to the carbo test strips. 2, urine is passed over strip, including the enzyme. And glucose binds to the immobilized enzyme is present, converting it into hydrogen peroxide or gluconic acid. You can write either of them, and causes a color change that shows the detection of glucose in urine. All right. So that is all for this question. So since we have answered this question, let's move on to the next one.